Our first speaker today is Jun Wei. He's in the oncogenomics section of the genetics branch. He got his bachelor's degree at the University of Houston and subsequently a PhD at Baylor College of Medicine. And he did postdoctoral training in Paul Meltzer's lab. And currently he assists Javed Khan, and he does the ongoing research in the oncogenomics section. So it's titled today, The Application of Genomics to Identify Diagnostic Biomarkers, Drivers, and Therapeutic Targets for Pediatric Cancers. June.
Yeah. Thank you, Terry, for the invitation to give this lecture. And here is an outline of what I'm going to do today. And first, I'm going to introduce you what is the success and the challenges of treating pediatric cancers. And the second topic I want to introduce to you is the genomics. And the third one is the next generation sequencing, which is the powerful genomic tools recently developed. And we are extensively using these tools to study cancers. And next part is I'm going to give you some examples to show what's the application of those next generation sequencing we can use on as a diagnostic tool. And we can also identify molecular targets for treating those cancers. And the last portion, I'm going to get into the precision medicine, which is the very important area of the medicine to use these genomic tools to make the precision medicine a reality. So this slide shows that the pediatric cancer, it's a very successful story. You can see in the 60s, which is depicted by the blue bars, the survival rate for all these three pediatric cancer is very low. But in the 90s, you see there's a dramatic improvement of the survival rate for the cancer patient with those cancers. However, in the past 16 years, you can see this is the mortality rate is decreasing. This is a lymphoma and leukemia. And this is other pediatric cancer. And you can see they are decreasing, but at about the year 2000, this decreasing start to level off. That means despite a lot of very aggressive therapy, and actually those therapies are very, not only aggressive, but they are very toxic. And but the mortality rate is still keep at a certain level. So that's the problem facing to the nowadays modern oncology in pediatrics. And if we look into the detail of those survival data, we found that the most patient cannot be cured with the pediatric cancers are metastatic, recurrent, and refractory disease. And if you look at the neuroblastoma, it's still like 30, 40s. And then Ewing sarcoma, the survival rate down to 20, 30s. And this is a rhabdomyosarcoma, metastatic rhabdomyosarcoma, and osteosarcoma. You can see all these high stage means they're metastatic and the recurrent tumor, the cure rate is still pretty low. This is the challenge of this pediatric cancers. Okay, so we have this phenotype, and how do we study those with the modern technology? And these slides show you that the relationship between genotype and phenotype. So you can see this is a caterpillar, and this is a adult butterfly. They look so different, right? But they share the exactly same genome. okay? So this is the same genome, but they have very different expression pattern and different proteome and different tissues and have different physiology. And all this, the summary of this is the phenotype, right? So this is also applied for the human, okay? Although human, we all have the same genome, but in our body, we have so many different kind of tissues. It's all coming from this same genome. So from the same genomes, we can have skin, different glands and airway, epithelial, tons, so on and so forth, muscle cells, neurons, you name it. This is a very, very different cells and the tissues, but yet they are all share the same genome. They are encoded by the same DNA, okay? So this complexity and the diversity is all resulting from this genome, okay? It's encoded in the genome. And this slide actually depicts the central dogma in biology. This is the information flow is from the DNA, and when DNA transcribed into the RNA, messenger RNA, and then is then translated into the protein, and the protein will is what we saw is the phenotype. However, this is not that simple. There's a lot of feedback loop back to, for example, protein can control the translation or stability of the messenger RNA, and also protein can control the genome to determine when the gene is on, when it's off, and how much is on, and how much the expression level is. So this is all the feedback loop. And recently, we also discovered that there's a group of RNA called microRNA, and they themselves is not coding. They do not coding for protein or other things, but they have very important regulatory roles for the transcription as well as the translation. So this is a very complicated system, but this is a very, it's a simplified version of the complicated system. But you can have some sense. The information flows is from the DNA to the mRNA, eventually to the protein. And this is the rough line to separate the study. This side, we studied the nucleotide acid of DNA or RNA, and we call it genomics. And this part, we studied protein, which is called proteomics. And in terms of number, we know that human genomes code about 30 billion, has 30 billion base pairs, right? And these 30 billion base pairs only code for about 25 to 30,000 genes. And this is actually it's a kind of surprising when the first human genome project finished. And they originally expected much more genes, but it turned out it's only 25 to 30,000 genes. However, there's a lot of, for each genes, they can have different splicing variants, right? So the estimation for this, all the splice variants is about 150,000. 3 billion. Sorry. Oh, did I say 30? Sorry. 3 billion base pairs. Yeah. And the small microRNA, there's about like 1,000 or so microRNA, and also there's about over 10,000 non-coding RNA. Those are all the regulatory elements to for the regulation of the genome. And on the protein side, it's estimated about 500,000 proteins. But on the top of the complexity, these 50 or 500,000 proteins, they can have a different form. For example, they can be phosphorylated, they can be methylated, so on and so forth. There's a modification groups towards that. and will change their functions and their activities. So that's the whole very like a 30,000 foot overview of this biological system. So it's a very complicated system, and the information flow is programmed by this genome, and it's very precise. And in terms of the timing, amount, and the place, spatial expression, it's all programmed in the human genome. And recently, the data shows that although only 1 or 2% is the coding sequence coding these 20 to 30,000 genes, right? But 80% of the human genome actually is transcribed. So before, people always think that most of our human genome is full of junk DNA. But now we realize these are not junk DNA. 80% of the human genome actually is functional. Okay. Yes?
Transcribed doesn't actually mean it's functional, though. Are you getting that from the ENCODE project? Because that was highly criticized.
Correct. That's from the ENCODE project, 80%.
Yeah.
Well, you can call it a crap, but there's incredible information from the ENCODE project, you know, and we understand a lot of things recently about the epigenome, epigenetics, modification, all these kind of things. It's all from the ENCODE project, and it reveals there's much more information than just the transcribing RNAs. Yeah, the complexity of this system is so huge. You know, we do not understand all of them. That's the reason before we think this is all junk DNA. Actually, you know, not necessarily. Yeah. So this is the first milestone of the human project, right? By 2001, I think after 10 years launch of the human project, and we have a first draft of the human genome. So this is like both journal has this debut for this completion of the human genome in 2001. And the genomic research in cancer actually take a huge advantage of human genome, of the information we got, because human genome gave us a complete map of genetic code that we can study with, and make a lot of research project much easier than before the human project. I just give you a simple example. You know, when I was a graduate student, we discovered a gene called cadherin 11. At that time, it's only 1.5 kb gene. Okay, it's very short. We want to sequence it. It took me almost 2 years to run the Sanger sequencing gel after gel to sequence that, 1.5 kb. But nowadays, you can sequence the human genome overnight, 3 billion base pair. So that's the power of the technology, okay? And the way we use human genome, this genome map, is we use this technology to also the information to identify the biomarker, and hopefully those biomarker is the driver of the disease, and then we can devise using the either drugs or synthetic compounds to interfere with this disease. That's the therapeutic target. And also, all this, hopefully, there will be low or no expression in the normal tissue, so you do not interfere with the normal function of our body. Okay, the challenge of this kind of a complicated disease like cancer is how to measure and detect genes and their product in a massively parallel way, because we have, so many, tens of thousands of genes. And this is really the genomics comes in, and the genomics is using the high throughput technologies to deal with this problem. And this is in hand with the technology in the computational technology. So we need to have enough computational power to do that. Before the computer have enough computational power, this kind of study almost is impossible, right? You can generate data, but you cannot process it. But now we have the power to process this kind of data. And these slides just show you the first generation genomic tools. This is called a microarray. And it uses all different kind of method to printing the probes, DNA probes, on glass slides or some solid surface so that these probes can be physically fixed on a particular spot, okay? And here I show you some technology, common technology. For example, you can use the mechanical. This is like all these little pens, they are like a quill pen. They dip into the solution and they print it on the slides. And this one is more advanced, is using electronic piezo. This is like the inkjet, right? We all use the inkjet printer. This is the same kind of technology. You can inkjet printing those probes on the glass slides. And later, there's a more advanced technology used like the lithographic mask. This is the same kind of technology to make the computer chip. So Affymetrix using this kind of technology to make their microarray. But this technology, at the very beginning, they have to generate a physical mask. In other words, it's the physical mask to let either light through or blocking the lights. This is a physical mask. If you want to generate a 20-something 20-mer oligos, you need to generate at least 20-something mask to do this. And this is very costly. And then later, they use this digital mirror, micro mirror technology, which is exactly the same technology as we are using in the digital projector. They can project the light to the place they want, they intended to finish this photographic chemical process. So this is a microarray. And if we have a piece of microarray with all these different probes, we know what the probe sequence is, and we can label the samples either from our health tissues or cancerous tissues with different fluorophore, and we'll put into the solution, hybridize into this array. And this hybridization happens to specifically the probe will specifically hybridize to the target. And then we can use like a confocal microscope to scan the slides to get the image. And the intensity of each spot will indicate the signal, the original abundance of the molecule in the original sample. This is the microarray, how the microarray works. So it's a technology by hybridization. But lately, we have a new tool, genomic tool. It's called the next generation sequencing. And in this technology, what we do is just basically it's a shotgun sequencing. So if you have a biological sample, either DNA or RNA, and this technology is first will do the sharing or fragmentation of this molecule, turn to a small different size. And then after size selection, you put the adapter, the common adapter, onto end of this DNA or RNA fragment, and you can sequence those in a massive parallel way. And then you align all those little sequences to the reference genome, reference human genome. And then you can reconstitute the original sequence from the sample. Okay, that's just the principle behind this next generation sequencing. And here is a real picture of the image coming from the next generation sequencing. This is a field all these little dots is one cluster of DNA strand, okay? And if we blow up this little square, you can see this is a very dense clusters of different DNA strands. So each spot is equivalent to one Sanger sequencing. So in the same time, that's the reason it's called a massive parallel sequencing. You can sequence hundreds of millions of those DNA. And the image I just show you, it's about this size on a whole flow cell, okay? So you can imagine, you know, with these 4 lanes of flow cells, you can acquire a huge amount of data in a very short period of time. Okay, this slide shows what kind of information we can get from this kind of technology, okay? And first we can get the point mutation. If we align all our reads, we compare to the reference genome, we said, hey, this is supposed to be an A, but it's a C. So we know there's a mutation happen here. And also we can find the indel, which is a small insertion and deletion. And all the reads here, it will be, in this case, will do not contain the sequence from this side, right? And if there's a bigger region of deletion, if it's a homozygous deletion, you won't see any reads at all. But if it's a homozygous deletion, you will see some reads, but not as many reads as the adjacent region. And again, if there's a gain, you can see there's more reads than it should be, right? And another thing is this kind of reads contains on the same molecule, it contains the sequence coming from the different part of the genome, in this case from the chromosome 1 as well as chromosome 5, which is unusual. That's the reason we can detect the translocation and the breakpoint. You know, all those kind of things, if we want to detect in the past, it's a pretty laborious process. You have to clone it out, you have to sequence one by one. It's very laborious. But now you can do everything very quickly. And then another thing is you can also detect the pathogens in your sample. For example, some tumor is caused by the pathogen, like the human papillus virus will cause the cervical cancer. And actually from the cervical cancer samples, you can detect this kind of pathogen, which is not a human sequence. This is on the DNA side, and the same with the RNA. And using this technology, we can acquire digital expression from the RNA samples, and we can also detect express the mutation, alternative splicing event, and the fusion events, and the RNA editing, and novel transcripts, and non-coding RNA. Okay. And the advantage of next generation sequencing technology versus the microarray is there's no need to prepare clones for the DNA fragments, right? And there's no need of prior knowledge of the probe design, because for all the DNA microarray, if you want to detect something, you need to know what you want to detect. Then you have to design specific probe for that thing. For example, if there is a fusion gene, which is not naturally happen, if you do not know, it's very hard to design some probe to catch that fusion part. Another advantage is sometimes even the microarray, you can have some indication of gain and loss of genomic material. But if it's a balanced translocation, for example, just 2 chromosomes join abnormally to each other, on the microarray, you cannot detect that event because it's balanced. But in the next generation sequencing, definitely you can detect such event. And it's because it's sequencing based, you can generate the base pair resolution data. So immediately you can know where's the fusion at, where's the mutation at. You don't need to, like the past, you have the map to the chromosome, try to do the linkage study. And with the technology getting mature and mature, and the sequencing actually has become cheaper and cheaper, and faster per genome cost. And this next generation sequencing is also can allow to acquire all different kind of information, use the same platform. You can, in the past, if you do microarray, you have to design different array, for the copy number array, SNP array, expression array, all different kind of different array. But for this next generation sequencing, you just need the same, just one sequencer. And depends what kind of library you make, you can get all different kind of information. For example, for the DNA side, we can get the whole genome sequencing, and we can use the whole exome sequencing, which use the probe just to pull down the coding region. And the methylation sequencing, you only sequence the places CpG island that is rich with the events of the methylation. And also you can do the ChIP-seq, which is pull down the transcription factor binding area to see where's this transcription binding specifically. And the RNA, you can do the messenger RNA sequencing, non-coding RNA sequencing. And this is all the information you can get. DNA side, as I said, is a copy number, gene rearrangement, and the entire genome or some novel methylome or epigenome sequencing. And we can also easily detect the damage mutations. On the RNA side, again, there's information for the gene expression of the sample and the chimeric genes, splice variant, novel transcripts, and the damage mutations. And all those we can to identify the biomarkers and understand the biology of the cancers, and hopefully we can identify the therapeutic targets. So here I gave you an example using genomic tools to deal with clinical cases. And there is a 4.5-year-old girl sought for second opinion from POB. And this is from Germany. And their concern is the questionable diagnosis, because this girl's cancer is not responsible as it should be as the diagnosis. And this girl has a 6 week of history of weight loss and reduced appetite and fever and abdominal pain. And on the examination, there's a big abdominal mass showing this MRI scanning. This is the mass, and also in the vena cava, there's also a mass. So the initial diagnosis is a Wilms tumor. Okay. However, when we run a microarray experiment, this is like almost 15 years ago, when we run the microarray gene expression profile, this is a PCA plot to show different diagnosis group. You can see this is a rhabdomyosarcoma expression. This is the Wilms tumor. This is a Ewing sarcoma. This is neuroblastoma. And this is the sample we're talking about. And instead of its grouping with this Wilms tumor, actually it groups with the neuroblastoma. So that's the reason we think this is not a Wilms tumor at all. Instead, we revised the diagnosis into the neuroblastoma. And since then, the patient was switched to a high-risk neuroblastoma treatment, including the stem cell transplant. And this girl actually doing pretty well after a year at that time. So that shows you how important this, what's the application of the genomic tools can help in the clinical application. And since then, we do not use microarray that much anymore. We use next generation sequencing. In this slide, you can see that the next generation sequencing, if we do whole genome, we can easily detect this kind of translocation. This is from the translocation happened from chromosome 2 to chromosome 13, PAX3 FOXO1 translocation. It is a hallmark for the rhabdomyosarcoma. So if you saw this translocation, you can very confidently diagnose this is rhabdomyosarcoma. And same thing with this case, it's a Ewing sarcoma, has a hallmark translocation between the chromosome 11 and the 22, EWS flying. And this is very easy also using the RNA-seq. If we look at the RNA, we can easily to detect those abnormal fusion, as shown here in this publication. So about 5 or 6 years ago, at that time, the field of pediatric cancer, there's a lot of sequencing project going, and the first result that come out from all those big sequencing project is those pediatric cancer actually has a very low mutation burden compared to the adult cancer. These are all the adult cancer from the melanoma, stomach cancer, so on and so forth. You can see it has a lot of mutation per megabase. But when you come down to the pediatric cancer, most of them has very few mutation. So the question at that time is there any enrichment for the actionable mutation at the relapse of those samples? And is mutation load similarly low at the relapse? And is there any clinical utility of this kind of study especially when the patient at the relapse stage? And what is the feasibility and the performing genomic guided precision therapy trials in the pediatric? So that's the questions we want to address. We designed this study. We call it Clinomic. And the Clinomic is using the state-of-art multi-dimensional integrated approach to study the pediatric cancer. This is study design. It's a pilot study, and we try to determine the utility and the feasibility of performing comprehensive genome analyses to identify clinically actionable mutation in the pediatric and the young adult patient. And in this study, we have 59 patients enrolled into this omic protocol at the POB branch at the CCR. And the time span is from 2010 to 2014. So the average age of the patient they recruit is about from 7 months, very young patient, to 25 years old. And it covers about 20 diagnostic categories. And we performed the comprehensive multi-omics, exome, germline, as well as tumor and tumor RNA sequencing. And in addition, we also performed the SNP array analysis on those tumors to get a copy number change information. So the first is what is the definition of actionable? Okay, the actionable definition for the germline mutation is a loss of function mutation or non-hotspot activating mutation of a cancer consensus genes or pathogenic or lack pathogenic mutations defined by the American College of Medical Genetics. Okay, at that time there's a 56 genes in this category. And in terms of the somatic mutation, our definition is genomic alterations that change the patient's diagnosis or may be targeted by FDA-approved drugs or in context of the existing clinical trials according to the NCI adult match trial criteria. So there's a whole criteria to define what kind of mutation is actionable or not, if there's any trial or drugs is indicated by those mutations. And here is the overview of the data generated from this study. And again, it's a multi-omics and it's integrated. The reason we said it's integrated is shown as following. So this part of data, you can see, this is generated from the RNA-seq. And from here, we can see this is the fusion gene detection. You can see all those blue squares indicate those samples has a particular fusion gene. That's a diagnostic. Also, it's a driver event, and some of them can be actionable. For example, if there is ALK fusion, like this RNBP2 ALK, because the ALK is a tyrosine kinase, so there's a drug directly can target this fusion. And this part is summarized of the DNA-seq and the RNA sequencing of the somatic events. This is driver actionable. That's including all the mutations in those genes. And the little plus signs indicate that this is a hotspot mutation. That means it's recurring and is proven to be disease-causing genes, disease-causing mutation event. And this is the DNA copy number. Also, we validated in the RNA sequencing, make sure it's not only amplified, also they are overexpressed. So this is the driver and the actionable. And here from the DNA sequence, we can also identify the germline mutation. And again, this plus signs, or this little plus signs is a report germline mutation that has known disease-causing function. Yes.
You see an increased number of percentage of the germline mutations in very young patients?
That's a good question. You know, the direct answer is not really. It's a little bit high, slightly higher in the pediatric cancer, but it's not that big difference. So currently, the detection rate of germline mutation in the cancer patient is about 10%. Okay, pediatric is a little bit higher, but 12% or so. But it's not that dramatic. Again, another thing is, you know, because this is a pediatric cancer, they happen very early. The hypothesis is most of those patients has germline mutation. That's the reason they can have no germline mutation. Yeah, the reason we didn't detect is not that's my opinion, is some mutation we just don't know if this is disease causing or not. Because this germline mutation, we only know, again, the information of those mutation is very limited. And it's very hard to distinguish what is germline mutation or private mutation. You know, by saying private mutation is if you just sequence me and you, everybody gonna have thousands private SNPs, which nobody has in the whole wide world. Okay, that's the reason we can do the forensic science, right? You can make sure this is your DNA, is not other people's DNA. But those may be just normal SNPs. They are not disease causing, but some variant, they indeed are disease causing. but we do not know. I just don't think we have a full knowledge of disease-causing variant yet on the germline space. And I want to highlight this. This part is because we can sequence those tumors with very precision, very good precision, and we can immediately identify the diagnostic fusion genes. For example, there's 4 samples I showed here. Actually, the original diagnosis was not correct. It's because we detected those hallmark fusions, and we were able to revise the diagnosis of these tumors. And this table shows that about 10% of those pediatric cancer patients, we can detect the germline mutations. And some of those are therapeutic. And you know, actually, this is an important thing, because if this germline mutation is detected in a patient, it's not a, that means this germline mutation might run in the family. So they have additional implication for the clinical meaning for the family, not only for the patient, but also for the family. So this is a very challenging and also the ethical thing. It's not that easy to deal with. This table shows that about 50% we detect the 30 out of 59 patients, we can detect the actionable somatic mutation. Okay, and again, some of you can see it's like the BRAF mutation, B600E, and we definitely have the drugs against those, and the same with the ALK mutation, and the P10, PI3, KCA, and all this mutation potentially is actionable. So in summary, using this technology, we demonstrate that the importance and the feasibility of performing this kind of multi-dimensional clinomic in the clinical setting in a real time. And 50% of the children, as I told you, has actionable somatic mutation. And 10% have actionable germline mutation. And the important performing the germline sequencing is to find the actionable therapeutic targets, for example, DNA repair genes. So you can use the drug to target this pathway. And also, I didn't show you the data, but we also observed that the increased tumor burden in the relapsed tumor. That the implication of using immune therapy for those relapsed tumor, because in the pediatric tumor, at the diagnosis, usually the mutation burden is low, but after the relapse, the tumor burden is higher. So probably we can use immune therapy to target those new antigens. Okay, and from this study, we envision that the precision therapy for the Fusion clinical trial is as following. So if there's a metastatic disease, in the current therapy, we treat all those patients the same way, okay? But if we have a way to use the genomics to detect the biomarkers, we can sort them into different categories. And some patients, if we know they have good signatures, and we can use standard therapy to treat them, and they're going to have better prognosis. But for those poor signature patients, and we can further to detect if they have a particular molecular events that can use as a target individualized combined therapies to treat those patients to improve their survival rate. And those events can including the amplification of the genes, mutation, translocation, overexpression, and alternative splicing genes. All these events can be targeted. And from that study that we now we are building up this program within the CCR. Hopefully, we can offer this kind of service to all the cancer patients enrolled in the CCR. So in the CCR, the characteristic of the CCR cancer patient is that they usually do not respond to the standard therapy. So they are referred to the NIH and the CCR to be enrolled on those clinical trials to find if there's any chance they can be responsive. So when the patient come to the CCR and they can enroll in different clinical protocol, and a PI, the principal investigator, they can order this next generation sequencing test to see what is the characteristic of the particular tumor. And all these tissues will be procured and go to the LP, the laboratory of pathology. They will do QC and the nucleic acid extraction, and they're going to send it to us to perform the next generation sequencing experiment. And the data will come out this experiment is germline data, somatic data, and we have genetic board or the tumor board to discuss what is the important change we detected. And those information will incorporate in the clinical electronic medical record and to provide this information for the PI to use for the precision therapy decision. And another thing is all those data generated can be deposited into the Beatrice system, which is the NIH system to treat as a research data. And in the future, we can mine those data to have new discoveries. So that's the idea behind this program. And in this program, we offer both exome sequencing as panel sequencing. So the exome is we sequence all the protein coding genes. This is giving us the breadth of this molecular profile of a tumor. So we can detect the mutations in dominant clones, novel driver mutation, and the actionable secondary incidental findings in the germline in the non-cancer genes. And this panel we designed is to get the depth because certain tumor, they might have clones in the tumor samples. If we sequence like that, the clone effect, it was diluted in the tumor, we may not be able to pick it up. But if we sequence it very deep, we can detect those events. And also this panel sequencing also can validate the exome generation, exome sequencing. And also this one can provide us copy number change and the LH, the loss of heterozygosity. So a lot of cancers, the two hits will happen is first they lost the heterozygosity and then inactivate another copy, the other copy of the gene. And also we can use this for the fusion detection. And this program, we also developed a data portal to present the data. You can imagine, as I alluded earlier, that the genomic data, it's not only, it's a large quantity, but it's very complex. So we developed this website to enable either physician, you don't need to have a programming skill to look into those data. And by the way, those data is so big, all the Excel spreadsheet, everything we deal with in text file, you cannot deal with those kind of data. You cannot even open those files. So to understand and to view those data can be very challenging. But this kind of a way, we can use this website to host all the data, and we can view those data very easily, even as a biologist. And this is the website, and we can use NIH credential to log in, and it's a secure data deposit, and it's access controlled. Every time you log in, you have a record who's logging in from where. And we have a interactive data viewing. So you can look at the QC of the data, quality control of the data, and you can look at the DNA sequencing data, the exome cancer panel, and as well as the RNA sequencing. You can cross-check. If I see the mutation in the exome DNA data, is this the same data in the cancer panel or in the RNA-seq? You can cross-check all those experiments. And there is some screen capture of that. For example, this is running statistics. This is each line represents one sample, one sequencing. And you can see this is all the total reads, how much we sequenced, and what is the property of the sequence. Are those reads are unique, or there are a lot of duplicates? And what's the coverage at 30x, 50x, 100x, so on and so forth. And also we do genotyping using those genotyping too, to make sure if we say these two samples from the same patient, and they should match genotypically, right? In the lab, it's very easy to swap sample or make mistakes. And this is the tool that we ensure that the sample is the sample we think what they are. And these are other QC tools. We have circus plot. Here we plotted the variant allele frequency in the sample. This track is the germline, the green track. This orange track is tumor. You can see this tumor is has lost the heterozygosity in the chromosome 1, 2. This is the chromosome 4. And also chromosome 6. Here, this is an indication of this is a pretty pure tumor. Okay, and here is the RNA coverage, and you can see this is a 5' UTR all the way to 3' UTR. So the coverage is pretty even. If you have a degraded RNA, you will see this curve shift like this, because every time we're using the 3' UTR probe to synthesize the full clone. And this is just show the coverage plot. And this is the hotspot coverage on those known hotspot cancer genes. What's the coverage in that? You can see it's very high coverage. In the panel is about like a 500x coverage. And this is a particular view for the mutation view. And you list all the mutations and with all different annotations from the public database, and also give you the variant allele frequency. You can see this particular tumor, the V allele frequency is less than 0.5, most of them. That indication there's a normal contamination to this sample. And this is another view of mutation view across all the patients. For example, here on the top, there's lollipops where shows that the EGFR somatic mutation was detected in the CCR patient. And down here, it's all the mutations detected in those large cohort study. For example, the TCG or ICGC study. This is like the aggregate of hundreds of thousands people, patients, the mutation status for this particular gene. So this is a very important tool for us. Immediately we can realize that, for example, this one is a hotspot mutation because we detect in a lot of patients in those cohort study. So in conclusion, I just show you that the integrated analysis of cancer genome identifies biologically relevant diagnostic, prognostic biomarker novel targets for the therapy. And those powerful emerging tools of the sequencing will determine the complete genome portraits of a cancer at the base pair level. So that will increase our understanding of the cancer biology. That will also help us to identify the key drivers and being able to develop the future novel therapies. And again, this is a teamwork, and it involves multiple different team from genetic branch, from bioinformatics, and the research protocol. This is a clinical supporting team, and also from the laboratory pathology. And all this clinomic project is heading by the Paul Melzer, which is the branch chief, and Javed Khan as deputy chief in the genetics branch. So I'm going to stop here and see if you have questions.
Questions? Yeah, for the pediatric cancers, you don't have a lot of mutations, so you wouldn't think that immune checkpoint inhibitors would do very much. Yeah, that's a very good question. You know, for the diagnosis samples, indeed, that is the case. But as our data and the other people's study, when it's relapse, usually their mutation burden increase dramatically. You know, 3 times, even sometimes 10 times. So in that case, you still maybe have the chance to invoke the immune response from this checkpoint inhibitor. So, but this is a active research field. Yeah. Yes.
For your cancer panel, what's the cutoff for your final for the various?
Oh, currently, for the whole exome, we report about 10% allele frequency, but for the panel, we can go to 5%. As a matter of fact, we can detect the lower frequency, but in order to have enough sensitivity and the specificity, we choose 10% or 5%. Actually, we can go down even lower.
Okay, that'll do it.
Okay.
Thank you.
Okay, thank you.
So we have a few announcements. One being that June will be leading the Genomics Core Tour on Thursday at 3, and many of you have signed up for that. So you go over to building 37, the second floor. I'll be sending you an email to remind you on Thursday.
If you the vestibule, which is outside of the elevator, of the south elevator.
Okay, so it's the vestibule by the south elevator. And the second announcement is next week, TRACO will be on Tuesday, not Monday. And then unfortunately, our other speaker today was not able to be here. So through the miracle of modern technology, we're going to be showing you the talk from last year.
So my name is Kieron Dunleavy, and I'm the clinical director of the lymphoid malignancies branch at the National Cancer Institute. And over the next 45 minutes or so, I'm going to talk about lymphomas. The first half of the talk is some general principles about the biology and treatment of lymphomas, and then I'm going to talk about some of the research that we have been doing in our branch, just to give you some sense of the translational work that we can do in lymphoid malignancies here at the NCI. So in terms of how common lymphomas are compared to other cancers in the US, in males, they're the 5th most common cancer type, and in females, they're the 6th most common cancer type. But the incidence of lymphoma is increasing, and there are a lot of reasons for that. First reason is probably because there is an increase in the elderly population. So as with really all cancers, as people get older, they have a higher chance of developing cancer. And that's true for lymphomas, particularly for certain subtypes of lymphomas. Also, certain diseases are associated with lymphomas. HIV is associated with it. If you're HIV positive, you've got about 100 times increased chance of getting the most common type of lymphoma, which is called diffuse large B-cell lymphoma, and about 1,000 times more increased chance of getting Burkitt lymphoma, which is a pretty rare lymphoma, but it's often EBV-driven and strongly associated with HIV. And when we talk about lymphomas, there are Hodgkin lymphoma and non-Hodgkin's lymphomas. And as you can see, non-Hodgkin's lymphomas are much more common. They make up 83% of all lymphomas, and Hodgkin lymphoma makes up 17%. So in the US, I'll focus on non-Hodgkin lymphoma a lot during this lecture. There are 75,000 cases of non-Hodgkin lymphoma per year. And as I said, the incidence rises with age, and that's the biggest reason why we're seeing an increased incidence of lymphomas. And shown here are SEER data showing you the period from 1992 to 2010 and the incidence of NHL in both males and females. And as you can see, there has been an increase in the incidence of NHL in both males and females during that period of time. But the good news is that there has been a reduction in death rate from lymphoma during that same period of time in both males and females, as you can see from these curves. So for most people who develop lymphoma, there is no known etiologic factor, but there are certain factors that put you at increased risk of developing lymphoma. So people who've got altered immunity have got an increased risk of lymphoma. Some of the examples of that are diseases where you have inherited altered immunity, like Wiskott-Aldrich syndrome. As I said, acquired altered immunity. Patients who have HIV have got a much increased risk of developing lymphoma. People who have iatrogenic altered immunity, such as people who have undergone a cardiac transplant or renal transplant or bone marrow transplant and they're on immunosuppressive drugs, these patients have an increased risk of developing lymphomas, and they have a particular type of lymphoma called PTLD, or post-transplant lymphoproliferative disorder. And people who have had certain types of chemotherapy are also at increased risk of developing lymphoma. At the NIH, we have a group who study Sjogren's syndrome, and interestingly, when we do blind biopsies of these patients, you could do blind lip biopsies, and you find that almost all of these patients have got evidence of mucosal-associated lymphomas when you look for it. So autoimmune stimulation is an increased risk for lymphoma. Acquired immune stimulation is Helicobacter pylori is an organism which you can get in the stomach, and there are certain lymphomas in the stomach that are associated with this. And actually, in some cases, if you eradicate the organism, if you eradicate Helicobacter pylori, you can see regression of the lymphoma. That's not in most cases, but it's in some cases. And hepatitis C and HIV are also examples of this. Our understanding of the link between chemical exposure and lymphoma is not very well established, but it would appear that certain chemicals like organic solvents, agriculture chemicals, and maybe others are associated with lymphoma. There's a lot of interest at the moment in the microbiome and microbial oncology, and while we've certainly identified some organisms that are definitely linked to lymphoma, I think that there are a lot of other infections that possibly cause lymphoma, but this has not been definitively established at this point in time. So of the infectious etiologies that have been associated with various lymphomas. Burkitt lymphoma, as I said, is a rare lymphoma. You see 3 different types of this. You see endemic Burkitt lymphoma in regions of the world like equatorial Africa. It usually occurs in children between the ages of 3 and 5. And this type of lymphoma, it often presents with jaw masses, and it is linked to EBV in 100% of cases. The other 2 types of Burkitt lymphoma are sporadic Burkitt lymphoma, which is the type that we see most commonly in the US, and then HIV-associated Burkitt lymphoma. And both sporadic and HIV-associated Burkitt lymphoma are EBV positive in about 40% of cases. Hodgkin lymphoma can be linked to EBV, and post-transplant lymphoproliferative disorder, particularly after kidney transplants and heart transplants, can be linked to EBV. Obviously, AIDS-related NHL is linked to HIV. There's an unusual leukemia lymphoma called adult T-cell leukemia lymphoma, which is caused by the HTLV1 virus. And this, again, is seen in distinct regions of the world, particularly in the Caribbean. And these patients develop ATLL, which is a very aggressive lymphoma and is largely incurable. And most of the therapies that we use for other lymphomas don't work for this type of lymphoma. Primary effusion lymphoma is seen almost exclusively in patients who have HIV, and it is linked to the HHV8 virus, as is multiple myeloma. Lymphoplasmacytic lymphoma is linked to hepatitis C. I talked about MALT, linked to Helicobacter pylori. And Mediterranean lymphoma is linked to an intestinal pathogen. So the most important thing, when a patient has lymphoma, has newly diagnosed lymphoma, the most important thing is the pathology and getting the pathology right. And really, the tissue needs to be looked at by a pathologist who's really expert in hematopathology and in looking at lymphomas and has seen lots and lots of these lymphomas before, because they're becoming, or they are, a very complicated group of diseases. So I think it's best if the person making the diagnosis is not a general pathologist who looks at all different types of cancers, but really focuses on lymphomas and hematological diseases. And for all cancers, there's a WHO manual, which is very helpful in making the diagnosis for pathologists, and it really lists all the different types of lymphomas and their different characteristics under the microscope, and also clinically how they present. And we're certainly in an era now where a lot of lymphomas should not only be diagnosed by how they look under the microscope, but also how the patient presents. So this is called a clinico-pathologic diagnosis. So when we have a new patient with lymphoma and we look at their biopsy with our pathologists, they always like to know about how old is this patient, what did they present with, what did their CT scan look like. So it's important for them to know that, because that helps in both establishing the diagnosis, and in doing that, they can request special tests on the tissue that will be guided by the clinical presentation. And then lymphomas are classified according to whether they're T cell or B cell. About 85% of lymphomas are B cell, 15% are T cell. Their location in the lymph node, the appearance of the cells, molecular features, specific genetic abnormalities, and clinical features, what the history was coming in and what disease sites the patient has. And if you look at the breakdown of non-Hodgkin lymphoma, the most common type by far is diffuse large B-cell lymphoma. That makes up about 40 or 45%, followed by follicular lymphoma. And after that, there are a number of different types, but they're really pretty rare, as you can see here. So for the most part, when we have lymphoma patients, they have diffuse large B-cell lymphoma or follicular lymphoma. As shown here is the lymph node, and depending on where in the lymph node the lymphoma arises from, it can be very, very different. For example, you can have marginal zone lymphomas, which arise in the marginal zone, and follicular lymphomas, which obviously arise in follicles. So this is important for the diagnosis. And most lymphomas are B-cell lymphomas, and you can correspond B-cell lymphomas to different stages of B-cell differentiation. Precursor B lymphoblastic lymphoma is, you can correspond that to very early B cells, see a little bit later, and then the main lymphomas that we see a little bit later. And I'll talk a little bit about this in relation to diffuse large B-cell lymphoma, because that's the common histology. And when the pathologist is looking at a new lymphoma under the microscope, they use immunohistochemistry to decide what type of lymphoma it is. The lymphoma has a particular appearance in terms of its morphology with H&E, but immunohistochemistry is very helpful. And this was something that really wasn't around 20 years ago. So, for example, if it looks like a diffuse large B-cell lymphoma, you would do CD20, BCL6, MUM1, CD10, and all of these stains will be very helpful in telling you that that is the definitive diagnosis. And likewise, there are T-cell markers, which can tell you it's a T-cell lymphoma. So immunohistochemistry is very important. Cytogenetics are done in certain cases. For a lot of B-cell lymphomas, follicular lymphoma and diffuse large B-cell lymphoma, they frequently have a 14-18 translocation, leading to overexpression of BCL2. So that's found in close to 100% of follicular lymphomas and in about a third of diffuse large B-cell lymphomas. The lymphomas that are associated with Helicobacter, a lot of those can have an 11-18 translocation. So if the pathologist is told this lymphoma is coming from the stomach, that's something that they might do to confirm that it's a MALT or mucosal associated lymphoid tissue lymphoma. And Burkitt lymphoma has a MYC rearrangement, so that's an 8-14 translocation. That's present in 100% of Burkitt lymphomas. So Burkitt lymphoma is a very easy lymphoma to diagnose under the microscope. It's got a particular, what's called a starry sky appearance because of the high proliferation of the tumor cells. But if you check for a MYC rearrangement, it's positive in 100% of cases. There's an unusual type of T-cell lymphoma called anaplastic large cell lymphoma, and that's got a 2-5 rearrangement. So the pathologist really decides to do these tests based on their suspicion or where they're going with the diagnosis. They're not done on every single lymphoma. It's really guided by how it looks under the microscope. So then when you have a patient with newly diagnosed lymphoma, how do you evaluate them? The first thing is you ask them what has been going on with them, what have they presented with. The most common presentation in lymphoma is having lymph adenopathy, and people usually notice this in areas where you can feel your lymph nodes. So they notice it in their neck, under their arms, in their groins. If you have lymph nodes in your abdomen, it really takes a long time before you notice them. So most of our patients, when we ask them, what have you been complaining of, they usually say, I noticed a lump in my neck or under my arm or in my groin. And these are usually caused by infection, but when these are caused by lymphoma, they usually don't go away with antibiotics, obviously, and they're also usually non-tender. Infectious lumps tend to be tender, whereas most patients who present with a lymphoma, if they have a lump, they'll tell you that it doesn't hurt at all and it isn't really causing me any discomfort or any distress. In the history, we ask specific questions about how people have been, obviously, but there are symptoms called B symptoms, and those are fevers, night sweats, weight loss of more than 10%. And those are important because they put patients into different prognostic categories. So depending on the lymphoma type you come in with, if you have B symptoms, that might indicate that you have a more aggressive lymphoma and it's associated with a worse prognosis than if you don't have B symptoms. Everybody who comes in, you should You should examine them, obviously. That's very important. And then do blood tests. All of the basic tests should be done. There's a test called lactate dehydrogenase, which is a liver enzyme, but it's a non-specific test of tumor bulk, and that's often elevated in aggressive lymphoma. So that's a test that patients should have done. Once all the and everybody who presents with lymphoma, they should have a HIV test done because of the association with HIV, and they should also have hepatitis screening done. They should have hepatitis screening done for two reasons. One being that hepatitis can be associated with lymphomas, and two, certain types of hepatitis can reactivate with chemotherapy and with rituximab, which is a drug that nowadays we give to all patients with B-cell lymphomas. Everybody should have imaging of their chest, abdomen, and pelvis, and that's called staging. And there are 4 different stages: stage 1, 2, 3, and 4. And stage 1 or 2 disease means that the disease is on one side of the diaphragm, and if it's 3 or 4, it's on both sides of the diaphragm. And that's really becoming less important. 20 years ago was important because if people had very early stage disease, they typically got chemotherapy, but then they got radiation treatment if it was in one particular location. But nowadays, radiation is used less and less frequently. So staging may be becoming less important. Almost all patients should have a bone marrow aspirate biopsy done. In the B-cell lymphomas, the most common ones, in follicular lymphoma, the bone marrow is involved in a very high percentage of cases, probably about 15 to 20% of cases of diffuse large B-cell lymphoma. And then other studies should be done as indicated. There's a lot of work in looking at the role of FDG PET scans in the initial diagnosis of lymphoma, so a lot of doctors will do these. And then, depending on where the lymphoma presents, for example, if you have a lymphoma in your brain, it's best to do an MRI scan. And certain types of lymphomas and certain presentations should have their spinal fluid checked. So if you have aggressive B-cell lymphoma and if you have involvement of what are called extranodal sites, sites that are outside the lymph nodes, or if you have involvement of the bones, or if you have a very high LDH level, there are certain criteria that suggest that it's really important to check the CSF, because the treatment that we give to patients, it doesn't really get into the central nervous system. It doesn't, for the most part, penetrate the blood-brain barriers. So you really want to know if a patient has involvement of the spinal fluid. It's rare to have it, but it's associated with certain clinical factors. So if a patient has those, then we would recommend that you check the CSF. And it's fine to interrupt me at any point if you have any questions, but just, or we can ask. I'm happy to answer questions at the end. This is the staging system that I told you about. I won't go into it too much, but there are 4 different stages, and 1 being just one lymph node area, and 4 being diffuse or disseminated involvement and involvement of extranodal sites. And as I said, the morphology of lymphoma is important. This is diffuse large B-cell lymphoma morphology. And typically, you see large cells. They're about twice the normal size of a lymphocyte. They have vesicular nuclei, prominent nucleoli, and are basophilic. And they're large cleaved or multilobated cells. And they may be rich in T lymphocytes. So over recent years, there's been a huge amount of interest in the tumor microenvironment. It might be something that some of you are focusing on, but it's really important in certain types of lymphoma. So not just the tumor cells, but the infiltrating immune cells around the tumor cells. And in follicular lymphoma and diffuse large B-cell lymphoma, if you look closely at what these infiltrating cells are and what their genetic expression is, you can predict how certain patients will do. So the tumor microenvironment is becoming very important in lymphomas, and we're starting to develop therapies that actually target the microenvironment, not just the tumor cells, but also the microenvironment. Okay, so what about the outcome of patients with lymphoma? How do they do? Are most of them cured? And for this talk, I'll focus on diffuse large B-cell lymphoma quite a bit because that's the most common type and the type that you see the most when you look after patients with lymphoma. So these are Kaplan-Meier curves, and they show on top overall survival and progression-free survival on the bottom. And you can see here that the interval is in months. So this is out to 100 months, which is quite a long time. And these are patients who, of all ages, who had diffuse large B-cell lymphoma, and they received the standard treatment, which is a chemotherapy regimen called CHOP, which is cyclophosphamide, H is hydroxydonorubicin or doxorubicin, O is oncovin, which is the trade name for vincristine, and P is prednisone, which is a steroid. So, and R is rituximab. And over the past 20 or 25 years, the most significant advancement in treating patients with DLBCL has been the addition of rituximab to CHOP chemotherapy. That has increased the cure rate by about 10 to 15%. So it's very important to give rituximab. And it's interesting because the adult world and the pediatric world in lymphoma are very separate. And the pediatric world has been very hesitant to give rituximab, and they really haven't been giving it to everybody up until a few months ago, where they were doing a big randomized study in children, and they were randomized to get rituximab or not to get rituximab, but they have recently shown a little bit earlier than they expected that there was a significant survival advantage to giving rituximab. So now everybody, adult and child, who has diffuse large B-cell lymphoma gets rituximab. But I think what these curves tell you, I mean, if you're a new patient with DLBCL, and these are, patients can be divided into low risk IPI, intermediate risk, or intermediate high risk IPI. It's called the International Prognostic Index, and it looks at your age, the stage of disease that you have, whether you have an elevated LDH, your ECOG performance status. So if somebody's really fit, it's 0. If somebody's not really fit and lying in bed, it's 2 to 3. And then the presence of extranodal sites. And that's what the low risk, intermediate risk, and high risk is. But these curves just tell you that with standard treatment, so all around the world, if you go into an oncologist with diffuse large B-cell lymphoma, you get R-CHOP. But you can see that for a significant proportion of patients, standard therapy is not adequate. So, you know, while most people have a long overall survival, a lot of people don't, and a lot of people progress, as you can see from the curve on the bottom. So our field has really been trying to improve the cure rate for DLBCL, and a lot of different strategies have been tried. What have people been doing? You know, they have said, well, we usually give 6 cycles of R-CHOP, so let's try giving 8 cycles, see how that works, and that has not been helpful. It's associated with more toxicity, but no increased benefit. They've tried therapy intensification. So R-CHOP is typically given every 21 days. So people have tried giving it every 14 days. In Germany, about 10 years ago, there were some studies that suggested that if you gave it more intensely, it was associated with a better outcome. But about a year ago, a group in France and a group in the UK did large studies of patients where they gave half the patients R-CHOP every 21 days and half the patients R-CHOP every 14 days. And there was no difference in both studies. And the patients who got it every 14 days actually had more toxicity. So that wasn't helpful. People have tried doing bone marrow transplants when patients go into remission. That's not been of any benefit. But they've tried giving maintenance rituximab. That's where people finish their rituximab and CHOP treatment, and then they continue to receive rituximab for 18 months or 2 years after that. And studies have, you know, again, half the patients have had maintenance rituximab, half had not, and there was no difference between the two groups. What are promising strategies? Well, are there better chemotherapy platforms than R-CHOP? The French developed a regimen called RACVBP, and in young patients who have a low IPI score, that was better than R-CHOP. But that was a very small proportion of patients who get DLBCL. And our group has been doing a lot of work with dose-adjusted EPOCH-R, and we're awaiting the results of a randomized study. we have a lot of novel agents that are based on the identification of new pathways in different types of lymphomas. And I'll talk about those a little bit, but I think that that probably offers the most promise for improving cure rate, is to incorporate some of these effective novel agents into upfront standard regimens like R-CHOP. But as in our field, it's really critical to develop novel agents and approaches in the context of the biologic subtypes and factors. So in that respect, historically, we always thought that DLBCL was one disease, that it was just one entity. And this is work that was done by the Lymphoma and Leukemia Molecular Profiling Project. It's a big project that involves a lot of institutions around the US and Canada. And it was led actually by Lou Stout's lab, which is based at NCI. And about 20 years ago, they applied gene expression profiling to cases of lymphoma. And so they looked at the differential expression of tens of thousands of genes in a large sample of DLBCL cases. And when they did this, they were able to break down most of DLBCL cases into molecular subtypes. So most of them fell into one of two molecular subtypes, a germinal center B cell-like type, or GCB type, and an activated B cell-like type, or ABC type. A small percentage of them fell into another type, which I'll talk about at the end of the lecture, called primary mediastinal B cell lymphoma. So this is a subtype of DLBCL, but if you look at its genetic makeup, it actually has a lot more in common with Hodgkin lymphoma. It shares about a third of its genes with that, and so it's really very distinct from the ABC or the GCB subtype. And so, you may ask, well, this is very nice. It's very nice to be able to genetically divide this disease into at least three diseases, two of them that are shown here. But is that helpful for patients? Does that make any difference clinically? And it does make a difference clinically. So this is a study that was published last year from a group in British Columbia. They looked at the outcome of over 300 cases of DLBCL. So shown here is progression-free survival and time in years. And you can see that this group of patients have been followed for a very long time. And they did something called a Lymph2CX assay. So conventional gene expression profiling is expensive. And this assay has been validated with gene expression profiling. It looks at the expression of about 20 genes, and there's close to 100% concordance with conventional gene expression profiling. But the advantage is this is easy to do and much less expensive. But as you can see from here, patients who had the ABC subtype had a much worse outcome than the GCB subtype. And just to go back to this, the GCB, ABC, and PMBL, all subtypes of DLBCL, but they're really distinct diseases, and they have distinct mechanisms of oncogenic activation. So the GCB subtype comes from a germinal center centroblast, the ABC subtype from a plasmablast that's blocked during plasmacytic differentiation. And PMBL is a totally different disease that comes from a thymic B cell. So it arises in the mediastinum. And if you look at the oncogenic characteristics of these three subtypes, they're very distinct. For example, GCB, which makes up the majority, you often see a translocation of BCL2. In the ABC subtype, there's constitutive activation of the NF-kappa B pathway, and that has led to the identification of many interesting targets that are currently being pursued in clinical trials. And PMBL has got activation of Janus kinase pathways. There's a lot of interest at the moment in oncology with immune checkpoint inhibitors, and immuno-oncology is a huge area. And this is a disease. There's an early study of about 20 patients, but it responds very well to immune checkpoint inhibitors. So PD-1 and PD-L1 are very important in PMBL. So just talk a little bit about the biology of different subtypes of DLBCL. So The most common subtype is the GCB subtype. So if you look at children and young adults who get DLBCL, they almost always have the GCB subtype, and the ABC subtype starts creeping in when people get older. So if you look at people over the age of 40, about 5 or 10% of them have the ABC subtype. If you look over 60, it jumps up to about 20 or 30%. If you look over 80, it's over 50%. So the GCB is the type that you see in both young and older people, but in pretty much all young people. And about 10% of those have got a MYC rearrangement. And I think it's interesting to think about that in the context of whether the lymphoma is GCB or ABC. So this is diffuse large B-cell lymphoma in general, but about 10% of cases have got a MYC rearrangement, but these are almost always of GCB origin. Whereas cases that have high MYC expression but do not have a rearrangement are almost always of ABC origin. About a third of cases have got a BCL2 rearrangement. Again, these are almost always of GCB origin, whereas cases that have got high BCL2 expression but without a BCL2 rearrangement are almost always of ABC origin. So in our field, one of the most controversial areas are the double hit and double expressor lymphomas. You hear a lot about those, and when patients call us and they want to come to the clinic and they're asking about what the best therapy is, they talk about double hit and double expressor lymphomas a lot. And double hit lymphomas are those that have both a MYC rearrangement and a BCL2 rearrangement. And then double expressor lymphomas have got high BCL2 expression and high MYC expression. But this distinction between double hit and double expressor lymphomas is really important in our field in relation to the cell of origin, GCB or ABC, because it's starting to help us decide what therapy patients should get. It's really important then to distinguish the double hit lymphomas from the double expressor lymphomas that do not have any hits, because pretty much all the double expressor lymphomas that do not have any hits are of ABC origin, and almost all the double hit lymphomas are of GCB origin. So we're starting to think more and more of DLBCL in the context of cell of origin and in the context of these rearrangements because they make up a high proportion of new cases. So the ABC subtype is associated with a worse outcome. So there's a lot of interest in that because we really need to augment the cure rate of ABC lymphoma. And the key characteristic of this subtype is that there's activation of the NF-kappa B pathway. So these are NF-kappa B target genes, and you can see that ABC is on the left, GCB on the right, and there's a lot more red on the left. So there's the NF-kappa B target genes are much more highly activated in ABC DLBCL than they are in GCB DLBCL. So NF-kappa B is highly overexpressed, and therefore it's a potential target in ABC DLBCL. And over the past 10 or 15 years, a lot of work has been done in understanding the mechanisms of NF-kappa B activation in the ABC subtype of DLBCL. So for about 15 years, we've known that about 10% of patients have got a CARD11 mutation. But recently, over the last 4 or 5 years, it's been identified that about 20% of patients who have ABC DLBCL have got a mutation in CD79A and B in the B-cell receptor. And about 30% of patients have got a mutation in MYD88. And both overactivation of the B-cell receptor and overactivation of MYD88 can activate NF-kappa B. So these are very important pathways in thinking about what drugs should one use to target ABC lymphoma. And we now have a lot of inhibitors of various components of this pathway or these pathways. I could probably put up about 20 to 30 drugs that are in clinical trials or in development that specifically inhibit, they're small molecule inhibitors that inhibit various components of this pathway. I'm not going to talk about all of those, but I'm going to talk about inhibiting BTK, or Bruton's tyrosine kinase. So it's a key enzymatic step in the activation of NF-kappa B in tumors that have got chronic active B-cell receptor signaling. So it really makes sense to think about inhibiting that. And you've probably heard about ibrutinib. It's a BTK inhibitor. There now are a lot of other BTK inhibitors. This was really the first one. And it covalently binds to the BTK active site. And this is a study that we were involved in, but the hypothesis was that, so in ABC, DLBCL, the BTK is a critical enzymatic step in a proportion of tumors to activate NF-kappa B. So therefore, inhibiting it might be an interesting therapeutic strategy. So to preclinically confirm that this was something that might be worth doing, both ABC and GCB cell lines were treated with ibrutinib, and different types of ABC cell lines were used. Those that were CARD11 mutated, and those that were higher up than CARD11, so they didn't have a CARD11 mutant. And you would expect that with ibrutinib that the CARD11 mutated ones would not respond to it, and that GCB would not respond to it. And that's exactly what happened. So the ABC cell lines that did not have a CARD11 mutation, so they were upstream of that. They responded well to ibrutinib. And that led to a clinical study of ibrutinib in relapsed and refractory DLBCL. So in this study, patients had to have relapse disease. They could be ABC or GCB, and they received single-agent ibrutinib until they had progressive disease. And as you can see from this waterfall plot, so orange is GCB and blue is ABC. Patients with ABC had a much better response. This is, those on the right had a shrinkage, those on the left had growth over 0 or below 0. But you can see that the drug is much more active in the activated B cell subtype. And in terms of responses, close to 40% of patients had a CR or a PR, and just one patient had a PR with GCB. So it wasn't very effective in GCB. So this really proved that ibrutinib does work clinically in ABC much more than it works in GCB. This was the survival, and again, you can see that the blue is doing significantly better than the orange, and that's the ABC versus GCB. This was one patient from the study, and with single-agent ibrutinib, you can see that they have had a very good response. One thing unique about this study was that when the results were being analyzed, a proportion of tumors had mutational analysis done to see which tumors exactly had the best response to ibrutinib. And as you might have expected, those that had a CARD11 mutation didn't respond. But the tumors with the best response were those that had both a CD79B and a MYD88 mutation. And there is now a study in newly diagnosed patients, and this study has actually been completed. Patients with the ABC subtype went onto this study, and they were randomized to get R-CHOP or R-CHOP plus ibrutinib. So we'll know in a few years' time if adding ibrutinib to the ABC subtype improves cure rate. There are a lot of other studies that are ongoing, which I haven't shown you here. One, adding lenalidomide just like this. That's an immunomodulatory agent that appears to be much more effective in the ABC than in the GCB subtype. So these studies will help us in the coming years to see if the ABC subtype is susceptible to adding one of these novel agents or small molecule inhibitors to standard regimens like R-CHOP. Just going to talk a little bit about primary CNS lymphoma when I'm talking about ABC. This is some of the work that we've been doing here. So primary CNS lymphoma, that starts in the brain for the most part. It can start in other areas of the CNS as well. And over 90% of these tumors are diffuse large B-cell lymphomas. Sometimes you can see other types. And in keeping on the ABC theme, these tumors, they resemble the ABC subtype, but they're not exactly similar, or they're not exactly the same. And broadly, you see activation of NF-kappa B. CARD11 is mutated in a significant proportion. Mutations of MYD88 are common, as are mutations of CD79B. And it's really interesting that shown here are MYD88 mutations in systemic ABC subtype, an ABC subtype called primary cutaneous lymphoma, primary testicular lymphoma, and primary CNS lymphoma. And while I think historically have thought of all of these as of ABC subtype and similar biologically, when we look at mutational status, they are very different. And you can see that over 90% of primary CNS lymphomas have got a mutation of MYD88 versus about 30% of systemic ABC lymphoma. And if you look at CD79B, it's also mutated in over 50% of primary CNS lymphomas. So most primary CNS lymphomas have got both a MYD88 and a CD79B mutation. And why is that interesting? It's interesting because what about using ibrutinib in primary CNS lymphoma? So for patients with primary CNS, although they're diffuse large B-cell lymphoma, they're treated in a very different way. And the reason for that is because the treatments we use in systemic lymphoma, like R-CHOP, they don't cross the blood-brain barrier. So for that reason, historically, this group of patients have received very different types of treatment. And they've received drugs or approaches that overcome not being able to cross the blood-brain barrier. So the two drugs that have been, or still are standard in this disease are high-dose methotrexate and cytarabine. They're actually not very effective drugs for DLBCL. We would never use them in systemic DLBCL, but they're used, they have been used a lot in this disease because they cross the blood-brain barrier. And cranial radiation has also been used a lot, and autologous transplant. But these modalities have been used because of really the failure of other agents to be effective in this particular disease. And this disease has been very challenging to institute optimal trials in. It's rare. It's difficult to biopsy patients. And a lot of these patients have got a very poor performance status, much more so than systemic ABC lymphoma. So we designed a new treatment for this disease. We took an effective regimen that is good for systemic large cell lymphoma, the EPOCH-R regimen, and we thought about it, and the drugs that did cross the blood-brain barrier, we included those, and if drugs didn't cross the blood-brain barrier, we substituted those for drugs that did. So etoposide does cross it, so we included that. Prednisone doesn't, but dexamethasone, which is also a steroid, does. Vincristine doesn't cross, but we don't really know how effective vincristine is in DLBCL and if it contributes to curability. So we left that out. Cyclophosphamide doesn't cross, but temozolomide, they're both alkylator agents, does cross and has been used in lots of brain tumors, so we included that. Doxorubicin doesn't, but Doxil, which is liposomal doxorubicin, does, so we included that, and we included rituximab. Ibrutinib, as I said, because of the high rate of MYD88 mutations and CD79B mutations, we really thought that would be a good idea in this disease, but we didn't have any information that it crossed the blood-brain barrier. Nobody had ever done studies or had ever done PK levels in the CSF to check that, so we included that as well. So we called this regimen dose-adjusted TEDI-OR and combined an agent that targets B-cell receptor signaling with agents that penetrated the CNS effectively. And this is the regimen that we used. And so we gave patients 6 cycles of this. And in the very first cycle, we gave ibrutinib as a single agent for 14 days to see if it was effective in this disease and also to do PK studies in the CSF and in the peripheral blood and assess if it was getting into the CSF and at what concentrations it was getting in, if this was effective or not. So, and what do we see? We've now treated 18 patients, but these were our first 2 patients, and we saw that it did get into the CSF for a significant amount of time and achieve meaningful CSF concentrations. The 2 top curves are plasma, ibrutinib, and its active metabolite. The 2 bottom curves are CSF, ibrutinib, and its active metabolite. And I'm not going to show you the PKs for the whole 18 patients, but this shows you that the drug gets in there at pretty good concentrations. This is over a 24-hour period. So we've treated 18 patients and have really had very good responses with the ibrutinib alone and with the dose-adjusted TEDIOR. I'll just show you 2 anecdotes from the trial. This was a patient who was 67. Back in 2010, she presented with slurred speech, nomic aphasia, gait instability. When she was worked up with various tests, an MRI showed a lesion in the left cerebellum that was biopsied. It showed diffuse large B-cell lymphoma, and she got high-dose methotrexate, had progressive disease on that, got whole brain radiation and with high-dose cytarabine, had a complete response with that, but then in 2014, she relapsed with left temporal lobe disease and also some disease outside the CNS. And actually, when she came to see us, her doctors were considering hospice at that point. So this is her MRI scan before and after she got 14 days of ibrutinib. So at the very beginning of the study, she didn't get any chemotherapy, just ibrutinib for 14 days, and MRI before and after. And you can see that this left temporal lobe lesion has regressed significantly after ibrutinib treatment. This was another patient, again, another patient with relapse disease who had a lot of treatment in the past. She had a mass in the area of the brain called the corpus callosum. And again, after 14 days of ibrutinib, she had a good response. So I'm just illustrating to you that this drug works very well in primary CNS lymphoma. And we've treated 18 patients with the TEDIOR, and we have a number of patients who were very refractory to other treatments and are doing well and are remaining in remission. She's in remission now for over 2 years after this. So just to finish up, I'm going to talk about another type of mediastinal B-cell lymphoma that affects younger people, especially younger females, and this is called primary mediastinal B-cell lymphoma. So mediastinal B-cell lymphomas come from a thymic B-cell. So we all have a thymus, and in younger people, the thymic gland is much more prominent than it is in older people. And classical Hodgkin lymphoma comes from a thymic B-cell, as does primary mediastinal B-cell lymphoma. And this just shows you that there's a spectrum of these B-cell lymphomas that come from a thymic B-cell with primary mediastinal B-cell on the right and nodular sclerosing Hodgkin on the left. And if you look at their morphology and immunohistochemistry, you can, for the most part, say that one is Hodgkin and one is PMBL, but there are diseases in between that are intermediate in terms of morphology and immunohistochemistry between the 2 entities. And these are called mediastinal gray zone lymphomas, and there's a lot of interest in these. I'll just quickly go through this. But the therapy of this disease has been controversial. Most patients who get this disease, they're females in their late teens, 20s, or 30s. And historically, the way that this disease has been treated is by giving chemotherapy followed by mediastinal radiation. But of course, as these patients are young females for the most part, and mediastinal radiation is a very effective treatment, but down the road, it's not a good thing. We now know from studies in children that if you get mediastinal radiation, if you get high doses of it, and if you look at patients 10, 20, 30 years later, a very high proportion of them develop cancer and ischemic heart disease. So for females, the rate of breast cancer is about 20% starting 10 years after the end of treatment. So this is a disease, because it affects younger people, predominantly females. You really want to think about that when you are selecting upfront therapy, because you might cure the patient, but you don't want them to get breast cancer in 20 years' time. So I'll just quickly show you this in the interest of time. So there are a lot of different ways of treating this, but most of them include mediastinal radiation. With Wyndham Wilson, my colleague, we did a study with dose-adjusted EPOCH-R, which I've talked a little bit about, and hypothesized that if we gave that, we would not need to give mediastinal radiation because it's a regimen that we believe is much more dose-intense. It's dose-adjusted, and I won't go into it again, but this disease, just like classical Hodgkin disease, they both come from a tiny B cell. They both affect young females predominantly, and they historically have been diseases that benefit from dose intensity. So we started our study and said that this treatment is going to be effective, and we don't think that patients are going to need radiation afterwards. And that would be a significant shift in how we treat this, because if you could take radiation out of the equation, that's a big deal, because then you eliminate the chance of these secondary complications like cancers, and particularly breast cancer, happening. So we treated 53 patients, and as you can see from these Kaplan-Meier curves, the event-free survival was high without radiation. It was 93%. Just 3 patients did not have complete remission with this approach, and 2 of them got radiation, and they're both doing fine. And one of them, she had a small residual mass left behind, and we actually resected it, and her disease never came back. And for our ASH meeting this year, we have updated these results, and now we've got over 90 patients. And the results are a little bit less. The EFS is about 90%. But we've shown that you can effectively treat this disease without radiation with this regimen. So I'm not going to talk too much about that. I think for the last few minutes, I'll just take some questions, because I think we have about 5 minutes before the next talk. Thank you.
Where do you see the treatment going? You seem to be coming up with lots of new drugs.
Yes, I mean, I think that just like in several other cancers, I mean, the treatment is changing very, at a very fast rate. I would say in aggressive lymphomas, particularly in Hodgkin lymphoma, the immune checkpoint inhibitors, there are 2 that we use, one called nivolumab and one called pembrolizumab. And these really have made a huge difference, especially in Hodgkin lymphoma, but also in primary mediastinal lymphoma, which is less common. But so that's one class of agents which I think are going to change the field significantly. I've shown you some results with BTK inhibitors. I think that's throughout a lot of lymphomas and leukemias like CLL, mantle cell lymphoma, DLBCL. Certainly, ibrutinib has been very effective, and now there are a lot of other BTK inhibitors. So that particular pathway, I think, and inhibitors of it are very, very effective. And then there are new transplant treatments. Some of our colleagues at NCI are doing a lot of work with something called CAR T-cell therapy, where they take T cells out of patients and they genetically modify them and then reinfuse them, and they have all different types of CAR T-cell treatments that target specific antigens on the tumor cells. So there's been a lot of work with CD19, which is expressed on B cells and in diffuse large B-cell lymphoma and in follicular lymphoma, and now they're working on other CAR T-cells at present. I think they have a new study targeting CD30. So I think there are examples of some approaches that are very promising and very effective. I mean, the difficult thing about lymphoma or the B-cell lymphomas is that the cure rate is very high. So we have very effective strategies at the moment. So these new strategies are very effective, but bringing them into the upfront setting is challenging, because it's not easy when, in some of these diseases, when you have cure rates of 80 and 90%, to bring these new promising agents into upfront studies. So that's the challenge. But I think that the field has changed a lot over the last few years, and I would expect will continue to change at this pace with the drug development that's happening at the moment.
Good progress.
Yeah.
What's the success rate of this center?
The,
What is the success rate of this bone marrow transplant? How many patients?
the CAR T cell treatment or the so that's still pretty new, and it depend in lymphoma. It depends on the type of lymphoma. I think the field is still trying to figure that out because, I mean, it's at the point of being commercialized at the moment, but up until 2 or 3 years ago, a lot of groups all around the U.S. were really developing their own type of CAR T cell therapy. There are different vectors that you can use, and none of these approaches are exactly the same. And for the most part, people in their studies included all different types of lymphomas. At the NCI, Jim Kokendorfer, he published a really nice paper looking at CAR T cell therapy in diffuse large B cell lymphoma. And the numbers were small. There were about 12 or 13 patients, but there were 2 patients who had a very long remission of, they were actually 2 patients with primary mediastinal B cell lymphoma, and they had had multiple lines of therapy before. And after they got CAR T cell treatment, they had a very good response. And at 3 or 4 years, when he published his paper, they were both still in CR. So I think over the next few years, we'll appreciate a lot more, where the sort of niche is for CAR T cell treatment, and which types of patients we should be really thinking about it in the upfront setting. And yeah.
Thank you, Kieron.
Okay, great. Thank you.